Hello, and welcome to another genetics tutorial session. In this session, we're going to explore specifically the condition called aneuploidy, and I shouldn't really say the condition because there are many types of aneuploidies. But if you're going along in order, the last session we discussed large-scale chromosomal aberrations or restructurings. Well, now we're ready to discuss what happens when there's a change in the chromosome number. And in aneuploidy, what that means is there's usually one too many or one too few chromosomes. In other words, instead of having a homologous pair, right, two copies of a particular chromosome like you're supposed to, you would have either 3 copies of that homologue or 1, and there are other possibilities, but that's the idea. One or a couple extra or too few gives you aneuploidy. Euploidy sort of means true full set. Aneuploidy sort of means not a full, complete, proper set. Okay? So in order to understand this, we want to get some terminology down, and we want to look at the human karyotype. So as I say here, a karyotype is basically it refers to the structure of the genome chromosomally, but we say a karyotype is a picture, basically. It's an arrangement of an individual's chromosomes after we've isolated them, stained them, and photographed them under a microscope. We can literally see, chromosomes are big enough, I think you've gathered, we can see those chromosomes. So a normal human somatic cell, meaning not sex cell, have a total, as I'm sure you guys know, of 46 chromosomes arranged into 2 sets of 23. That's the diploid number, and algebraically, we say that 2n, because n is one full set, equals 46. I'm going to write it later, but that means that n, one full set, equals 23, and one of those sets, I think it's clear, but make sure, one of those sets you got from your mother, the other set you got from your father. And I'm speaking as if we're talking about humans, and we are, but this general idea applies to eukaryotic diploid organisms. Okay? So, as you guys also know, while most of the chromosomes are shared between the sexes, in humans and mammals in general, there is chromosomal difference between males and females. So the last set, we have decided to call them the last set of chromosomes, are the sex chromosomes, right? They're homologs, but they're the only homologs that are appreciably different from one another. So normal males have an X chromosome and a Y chromosome. So we say that they are XY. And normal females, as you guys know, I think we've discussed this earlier, have 2 X's, so they're XX. They're called the sex chromosomes because they act in sex determination. In other words, why will you develop as a male or as a female? So there's 1 pair of sex chromosomes and 22 pairs of all the other ones, which are known as autosomes. Autosomes really means not sex chromosomes, the regular other chromosomes. So there's 2 ways to look at this. They both mean the same thing. You have 2 complete sets of 23, right? Another way of looking at that is you have 23 pairs, right? So 23 homologous pairs. So you should have 2 chromosome number 1s, 2 chromosome number 2s, 2 number 3s, etc. If you look at it from the point of view of the pair, again, you got 1 from mom and 1 from dad, right? And just by convention, they have been named, I should say, numbered and named, just from the biggest to the smallest. So the largest chromosome is number 1. They're arranged in the karyotype. I'm going to show you a picture in a moment. And we just put them in order of size, and that's how they got numbered. And then particular banding patterns based on the stain get associated with these chromosomes so we can pick them out rather easily. Now, that's the normal situation, right? We're talking today about what happens if things change, so often changes in the normal chromosome number can have phenotypic consequences. Sometimes these are dire, really extreme. Sometimes they're not extreme at all, and we'll see why this happens. But first, let's look at a normal human karyotype. So when we write a karyotype, the shorthand is we say how many chromosomes are there total, and then as we'll see, if there's a problem, we'll write that in too. But there's no problem here, so we just say 46. Let's just quickly look at the salient features here. You'll notice that each chromosome, so let's look at number 1, the biggest one, there's a pair there, right? Every normal human being should have 2 of those, and there's a definite banding pattern. You can see the bands here. It's a little difficult to see exactly what the pattern is, and you have to really be an expert to be able to say, oh yeah, look at that, that's chromosome 1 just from the banding. But note that they do have banding patterns, and this has to do with how they take up certain stains. But the real thing to look for here is the fact that whichever one you look at, 1, 2, 3, etc., 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 there's a pair at each location. If you were to count all of these up, there would be a total of 46 chromosomes, right? So maybe it's a good thing to write on here. 2n equals 46. You feel free to count them if you want to. There are 22 pairs of autosomes, right? And this last pair is the sex chromosome pair. We separate them because this individual has an X and a Y, which means this individual was what? This is a karyotype from a human male, right? So he's XY. If you wanted to, sometimes they'll write 46XY because since the sex chromosome pair is sort of a special pair, sometimes that'll be written to the karyotype. But it's pretty wild and cool that we can actually see in such detail. Chromosomes are large, compared to some other microscopic structures. And there is a perfect, beautiful example of a normal human karyotype. Okay? But let's see what happens if things change. So firstly, we need to get down some general terminology, and I sort of implied this, but we want to be very clear about it, relating not just to this lecture, but this session, but to other ones. So when an organism has either one complete set or multiples of one complete set, we say that they're generally euploid. I used this term briefly before. Eu means true, and it means that these sets are complete. So we're used to thinking of diploid creatures, but let's start from the beginning. Bacteria are monoploid. They only have one set of their genes, right? Always. So bacteria are monoploid euploids. They're still euploids. It's a complete set. Eukaryotes like you and me are diploid, which I think you've thought of it, but diploid simply means di is a prefix that means two, right? So bacteria are monoploid, one set. We are diploid, two sets. But the point is, both us and bacteria are euploids, and other organisms that we're going to meet in a different session not only can have one set or two sets, they can have 3, 4, 5, 6, 7, 8, 9, 10. You can keep going. So anything that's a multiple of n, an exact multiple, is a euploid. Contrast that with aneuploidy, which is what we're focusing on in this session. This is not a difference in complete sets. It's, as I said, one or a few chromosomes present in either too many or too few copies. And algebraically, how would we write that? So the most common forms of aneuploidy is having one extra or one too few chromosomes. So if you had one too many, for example, if you had Down syndrome, we would say you have 2n plus 1. So the total amount of chromosomes there would be 47. 2n is 46, plus this extra one, you got 47, right? Or if you're 2n minus 1, you're missing one member of a homologous pair. I think obviously your total chromosome number would be 45, all right? So that's aneuploidy. That's what we're focusing on here. Not complete sets being changed, but just one chromosome here or there missing. Now, that makes it sound like, I should say missing or extra. Makes it sound like it's not important, but as we're going to see, it can be extremely important. So, again, just to get down some more terms, and we want to also understand how could this happen? What is the cause of aneuploidy? So terms, oops, sorry about that. That doesn't look nice. Terms and causes. Just a couple of terms. If you have one or more extra chromosomes, And this even relates to the other session, or even large extra chromosome segments like a duplication, right? We would say that you're hyperploid. This is a general descriptive term, simply meaning you got too much. It's general. It can be from a very small part too much to a whole chromosome too much, which is what we're talking about in aneuploidy, to a couple of chromosomes too many. You're hyperploid. The opposite of that is if you're lacking one or more chromosomes, or again, just a deletion like we talked about in the last session, we would say you are hypoploid in general. All right? Now, what causes aneuploidy? Almost always it's caused by an event called non-disjunction. Non-disjunction is the failure to separate, failure of chromosomes to properly separate and segregate during meiosis. All right? Now, it can occur in either parent, and it can occur during meiosis 1 or meiosis 2 with, I say, varying specific results. I had the artist make a couple of pictures. Let's just check them out quickly so we can understand how aneuploidy can happen and how it can come. In other words, the parents of aneuploid offspring usually are not aneuploid themselves, although it is true that if you are aneuploid, there's a good chance you will give birth to aneuploid offspring also. But here's a picture. We called it non-disjunction 1, not one sequentially, but we're going to have this one and another one, which we call non-disjunction 2. Check out what's happening here. We're only looking at one chromosome pair here, right? One homologous pair. If this was a human cell, there would be 22 additional pairs there. But what should happen during meiosis 1, which is this arrow right here? The homologous pairs, each homolog should separate and go into a new cell, right? So it should have one chromosome going into this cell and one chromosome going there. Well, they failed to separate. Both members of the pair went into one of the cells, and the other cell got what? Got nothing. So we've already got a problem, and if non-disjunction occurs at meiosis 1, all the gametes resulting from this will be problematic. All right? Because look what happens. In meiosis 2, remember what occurs. Sister chromatids get pulled apart. So we end up with gametes. Now, in this case, I'm saying that it happened in the mother, so these gametes are the eggs, but it could just as well be the father. How many copies of a particular chromosome are supposed to be present in the gametes? There's supposed to be one, right? But because of the non-disjunction, these two gametes got 2 copies, and these two over here got no copies. And what we're saying here in this picture, if its sperm comes along and fertilizes this egg, and we're assuming the sperm is normal, it's bringing its 1 copy, right? Now, it's expecting to encounter an egg that has 1 copy, thus making 2. But if it encounters this egg that already has 2 and adds its 1, right here, we're going to end up with a zygote that has 3 copies, and that is an aneuploid zygote. Let's introduce the term trisomy. If you have 1 extra, if you're 2n plus 1, you are a trisomic, or you have trisomy, all right? So both these individuals resulting from the gametes that got an extra copy now have 3 copies, and they're trisomic. On the flip side, the sperm comes over here and adds its 1 chromosome to a gamete that had none. Well, the end result is that that zygote now only has 1, and another term we can introduce is that that zygote is monosomic. It's 2n minus 1, and you can understand these prefixes, right? Mono means 1, tri means 3. So if you have monosomy, you're 2n minus 1. If you have trisomy, you're 2n plus 1. And these are both examples of aneuploidy, right? But let's also just see, because there are test questions that might go to this, and you might have to draw it out. If non-disjunction occurred during meiosis 2 instead of 1, what would be the result? How would this affect things? Well, meiosis 1 is normal, so as we said, one member of each homologous, you have your homologous pair, and the homologs separate, one going into each cell, right? So what we're saying is over on the right-hand side here, right, all over here, everything's going to happen normally, right? The sister chromatids separate. We end up with gametes that have one copy just like they should. And when they get fertilized, we get proper normal diploid offspring. But look what happens when non-disjunction occurs in meiosis II. The sister chromatids are supposed to be separating, right? But they're not separating. Or I should say they're not segregating properly. Both of them are ending up in one gamete, which is an aneuploid gamete, and none of them are ending up in the other, right? Now the principle is the same as before. If a sperm comes along, fertilizes the egg that had nothing in it, we're going to end up with a monosomic. If it fertilizes the egg that had two in it, we're going to end up with a trisomic. The difference is, I mean, you know, just for technical purposes, if it occurs in meiosis II, half the resulting gametes will be normal and half of them will be aneuploid. But the point is, if someone's aneuploid, you can't really go back and trace it and see where it happened necessarily. But we need to understand that these are two different ways that it can happen. All right? The result is, in either case, sometimes you can end up with monosomic zygotes, and sometimes you can end up with trisomic zygotes. All right? So that's the idea of non-disjunction. You should definitely be familiar with that. It's going to come up one way or another, most likely on a test question. Okay? So that explains how we can get aneuploid individuals. Let's talk a little bit more about what actually happens and some more terms and you know, the phenotypic consequences of this situation. Okay, well, we'll go over these terms. I just introduced them, but we want to focus on monosomy and trisomy. But there is something called nullosomy. Now, look, it's almost always lethal. You don't see living people that are nullosomic, but it's good to know the term. It's 2n minus 2, and that means both copies of a particular chromosome pair are missing. All right? This is fairly rare, but you should get the idea. Null means sort of nothing, right? Zero. If you're nullosomic, you're missing both members of a pair, and you absolutely can't live under any circumstances without at least one member of a homologous pair. Okay? But you should be able to mathematically know 2n minus 2. And the algebra is a little bit limited here because you could say, well, how do you know the two are from the same pair? The only way is because I'm telling you. Monosomy. We just saw how that can come about, 2n minus 1, right? So one member of a homologous pair is missing. Now, it turns out, and we've talked about this, or you have a little bit before, the proper dosage of chromosomes in general is 2, right? If there's 1 or 3, that's no good. We call this a dosage problem. You might think, well, can't the body just adjust to that? Unfortunately, not. Through evolutionary time, if there's too much, there's overexpression of genes. If there's too little, there's underexpression, and both are generally problematic. So if you're monosomic, this is usually a fatal condition. Now, when we say fatal, it's a little strange. I don't mean someone's born and they say, oh, you're monosomic, you're going to die. It means that this is not a viable situation. A zygote can't even come into existence in most cases that's monosomic. It would lead to a spontaneous abortion if it came about a miscarriage, right? But there are some exceptions, and by the end of this talk, we'll understand why. So one exception involves, and this is not coincidental, the sex chromosomes. If you just have one X and nothing else, right, you should either have XX or XY. We say XO, the O just meaning nothing is there. We call this Turner syndrome, and there are people that have Turner syndrome. They're alive. They're pretty much, okay. They look to be female, but they tend to be sterile, so that's a problem. And they have a variety of physical abnormalities, which is not I don't like to use the word abnormal, but its true meaning just is not normal, right? Not the norm. So, in other words, there are characteristics of these individuals that you could tell they have Turner syndrome. And the frequency of this happening is about 1 in 2,500 female births, so fairly low frequency, but if you think about it, that means there's going to be quite a few people around with Turner syndrome. This is an average, though. I just want to say the likelihood of this happening increases dramatically with the age of the mother. So this is a sort of average of everybody. If you're 21 years old, the likelihood is a lot less than this. If you're 45 years old, the likelihood is a lot greater. Okay? Turner syndrome. And I have a karyotype of someone with Turner syndrome. So let's check it out. Notice, I won't go on about it, but all the chromosomes are supposed to have 2 members, right? We should have homologous pairs. So if you were to look at all these, you'd say, yes, 1's okay, 2's okay, et cetera, et cetera, et cetera. But when we get here to the end, all we've got is 1 X chromosome and nothing else, right? And if you count these up, you guys, this is an example of what? 2n minus 1. And the minus 1 is missing in the sex chromosome pair. So the way we would write a Turner syndrome karyotype is 45, because if you were to count all of these, you can do it, but it should be obvious. 1's missing, there's 45 total chromosomes there, right? And we write XO because that tells us where the problem is. So this karyotype is 45XO. And an expert, guys, this is why they do something like an amniocentesis. They do an amniocentesis, look at the karyotype and say, this fetus has Turner syndrome. Now, what to do about that is a separate idea, if anything. Or sometimes, it's just useful to know this is how they first understood what this condition was, by doing a karyotype on someone who had Turner syndrome and saying, aha, look, they're missing a sex chromosome, they're XO. So I guess there's a couple of useful things you can do with a karyotype. But it's unlikely probably that a test question would say, here's a karyotype, tell me what this person has. But that wouldn't be a bad one, to tell you the truth. So you should be able to be familiar and comfortable with these karyotypes. Okay. What about trisomy? We talked about this. This is 2n plus 1, right? We have 1 extra chromosome. Instead of a homologous pair, we've got 3 copies at that place. So dosage problems still exist. It's apparently pretty much just as bad to have too much as too little, although it's probably worse to have too little. But just like monosomy, trisomy is usually fatal, and again, not fatal like you're going to die. You just don't see these because they abort. But there are several exceptions in this case, and not just with the sex chromosomes. So we'll just take a look at a couple of them. By far, the most common trisomy we see is Down syndrome. T21 is just short for trisomy 21, meaning these individuals have 3 copies of chromosome number 21. As you guys know, this is a common cause of mental retardation. Down syndrome individuals also have a very distinct facial appearance. It's very unpolitically correct. In the old days, they used to refer to this appearance as a mongoloid facial appearance. Don't use that word anymore, but I wanted to mention it because you may have heard it, and if someone says it, a mongoloid, they're referring to someone with Down syndrome, but very unpolitically correct. But the idea was because you guys have seen pictures, seen people with Down syndrome, whatever race they are, they have certain facial appearance that somebody thought looked like someone from Mongolia. I don't know. But that was the idea. And there are other health problems, often to do with the heart and other issues. Down syndrome people, many of them are very high functioning, can live perfectly reasonable lives. Usually the life expectancy is a little bit shorter than the average normal person. And Down syndrome is relatively common, 1 in 700 births. But again, remember, this is an average. And I won't keep saying this, but the probability of any aneuploidy always rises dramatically with the age of the mother. Okay? So this is, we said it, that's an average. If you're 21, the chances are a lot less than 1 in 700 that this will happen. Now, 2 other syndromes that exist, in other words, there are human beings in existence that have these syndromes, although tragically, usually not for very long. We have Patau syndrome, which is trisomy of chromosome 13. This is a lot more problematic. In fact, I would say trisomy 21, you guys, is the only autosomal trisomy where you normally would see an adult human being that's functioning, living, that has it. Patau's and Edwards syndrome, mostly these people die as very young babies. There are some exceptions, but that's the rule. So in trisomy 13, there are lots of abnormalities. Many people have all of them. You don't have to, but there's usually a mental deficiency that's fairly significant. Deafness is common. Polydactyly, which means having an extra digit. Cleft lip and cleft palate. Lots of cardiac anomalies. Guys, in other words, this is a bad syndrome, all right? Many individuals that are born with Patau syndrome die very, very quickly after they're born. It's a very tragic event. Now, luckily, this is less frequent, 1 in 20,000 births. I don't want to go deeply into this, but part of the reason is because a lot of trisomy 13 fetuses probably do spontaneously abort, and the ones that actually end up getting born, we just said, usually die relatively quickly. So that explains, at least partially, why there are fewer births that have this. In theory, all the trisomies occur at the same rate in terms of making zygotes. Hold that thought. Edwards syndrome, trisomy 18. There are a variety of organ malformations. Again, mental deficiency. I know it's vague, but a lot of problems. The statistics I found were that 90% die within 6 months of birth. A large percentage die earlier than that. There are some people who are able to survive with Edwards syndrome. It's not the rule, though. It's the exception. 1 in 8,000 births. So you can see from this that out of these three, Down syndrome is by far the most common and the most reasonable in terms of someone living a life. Now, here is a karyotype from a Down syndrome individual. Now that you're an expert at karyotyping, you should be able to look right around and say, bing, look at that. This should not be the way it is. There's 3 copies of number 21 there. There's 2 every place else. And by the way, is this a male or a female with Down syndrome? 2 X chromosomes, this is a female, right? So that's not particularly relevant, but I wanted you to just check out the karyotype. So we write down here that this Down syndrome individual is got a karyotype of 47 total chromosomes, right? Again, you can count them, but there's an extra. And the problem here is an additional 21. So that's the way we usually write it. 47 plus 21, all right? Not meaning plus an additional 21 chromosomes, meaning there's an extra number 21, okay? You could check out every other pair as normal, meaning they're a pair, but we've got this trisomy here at 21, okay? And I also, just to get used to these things, I found an Edwards syndrome karyotype. So we would call this 47 plus 18, and right here are the 3 number 18s responsible for this very severe condition, okay? This karyotype's a little more difficult to see. The chromosomes are not, whoever did this had a little harder time getting it to look good. But if you look around, you'll see that everything else is a pair. You'll see this individual is a male. Again, just to get practice, it doesn't really matter. You're just as likely to get Edwards syndrome if you're male or female. But here's the problem. 3 number 18s. Okay? Karyotypes are interesting and fun to look at. And I don't mean Edwards syndrome is fun, but getting to see the actual chromosomes. So much of genetics, you really can't see anything, right? Okay. Now, here's the thing. There are lots of examples involving the sex chromosomes. There's something special about the sex chromosomes where we can tolerate trisomy, and as we already saw, monosomy with Turner syndrome. And there's a reason. We'll get to it in a minute. But let's just check out some of these examples. XXX, so having an extra X chromosome. This is called triplo X syndrome. Some people call these super females, usually in fruit flies. But interestingly, guys, how to write this isn't the interesting part. Your 47 XXX, that's how we would write the karyotype, right? But generally, these are phenotypically normal individuals, and I guess I should write it, these are females. It turns out, in the beginning of the course, in a different session, we talked about, and I think you've already talked about the fact that having a Y chromosome really is what makes someone appear to be male. So that's why these are females. They have an extra, and it doesn't really seem to be much of a problem. Interesting. Again, hold on to that thought. All right, Klinefelter syndrome. This is someone who's XXY with the sex chromosomes, so they're also trisomic, right? These are all trisomic examples. 47XXY, but since they have a Y chromosome, they're going to look male. Now, Klinefelter males have a characteristic appearance. Again, my policy is I don't show you pictures of people, but you can look it up in your textbook or on the internet. Some of these males also exhibit some female secondary sexual characteristics like enlarged breasts, etc., because of the extra X. There is some sterility involved here, and sometimes mental impairment. The reason I say sometimes is because Klinefelter males have a wide range of mental functioning. Some of them pretty much normal mentally, so I don't want to malign anyone with Klinefelter syndrome. 1 out of 500 male births, but remember what this means. We say male births because these individuals appear to be male. So the way they collect the data, they say, okay, out of all the males that we see, 1 in 500 seems to have this syndrome. It's easier to keep track of it that way. What about XYY? You know, there are a couple of names for this syndrome, but they're not in popular use, so it's just XYY syndrome. A karyotype of 47XYY. Again, these individuals are male. They have 2 Y chromosomes. They have a generally tall, thin appearance. It's not ridiculously obvious, but an expert will be able to say that person has XYY. Sometimes there are mental irregularities, and I don't want to go into great detail about this, but there was an idea early on when we understood what XYY is, that these individuals might produce extra testosterone, which could lead to aggressiveness. This has never been borne out, but there's still studies that imply, or there's people that imply, that individuals that are XYY might be overly aggressive. I want to be clear about this. This has not been proven. But there was one famous study done in England, I think in the 70s, that implied that there were a greater proportion of XYY individuals in it was a facility for I guess what you would call criminally, mentally disturbed individuals. So take that for what it's worth. Plenty of XYY people are just pretty much fine. And remember, I just wanted to, this is back to monosomy, but I just wanted you to remember Turner syndrome is a sex chromosome abnormality, right? 45XO, that's a monosomy. And the reason I bring that back up here is because we're seeing all these examples of both trisomy and monosomy that have to do with the sex chromosomes. So let's look into why that is. I say that sex chromosome abnormalities are generally less harmful than autosomal ones. This is because of the fact, guys, this is something you probably already have talked about, but the proper dosage, the only time this is true of the X chromosome in humans is 1. 1 X chromosome gets turned off in any human that has more than 1. So for whatever reason, autosomes, the proper dosage is 2. Deviate from that, there's going to be a problem. There's no mechanism for doing anything about it. However, the idea is, since normal males have 1 X chromosome, normal females have 2, there has to be a mechanism that's evolved to deal with this dosage difference. And there is. It's called X chromosome inactivation or dosage compensation, right? So any individual with more than 1 X chromosome will turn off all of them but 1. And a turned off X chromosome is called a bar body. It gets heterochromatic, it gets all balled up, and you can see it in people's cells. So a normal human female has 1 bar body. Just quickly to make sure we understand this, a triplo X female, these are good test question type things. She would have 2 bar bodies, right? A Klinefelter male will have a bar body. That's interesting. Normal males, like myself, I don't know if I'm normal, but genetically, I don't have a bar body, right? I've got 1 X chromosome, and that's what I need, so that's it. But this one, or one of the 2, will get inactivated. So a Klinefelter male has a bar body. This person has no bar bodies. It's just, however, there's even examples of individuals that have 4 X's. They would have 3 bar bodies. And they live, and they're pretty much okay. Okay? So how many bar bodies does a Turner syndrome individual have? It's a female, but she has none. So this is interesting. Turner syndrome and Klinefelter syndrome helped a lot in understanding sex determination, because Klinefelter's individuals look male but have a bar body. Turner syndrome individuals look female but lack a bar body. And this was one of the things that led us to understand it's really not the amount of X's you have that makes you male or female. It's the presence or absence of the Y chromosome. All right? At least, now, this is true in mammals generally, definitely in humans. There are a lot of different sex determination systems in birds and other animals that gets out of hand. But this probably should have been discussed in an earlier time, and I know in our sessions it's been discussed way back toward the beginning when the idea of chromosomes was brought up. So I have a karyotype for you of someone with Klinefelter. You should be able to pin it out very easily that the problem here is with the sex chromosomes. There are 3 of them. But the sex chromosomes are great because you can so easily tell, which one's an X and which one's a Y. All the other groups, the homologs look identical, as well they should in autosomes. But here we see, wow, there's 3 of them, plus it's an XXY. So we can tell right, I could just show you this karyotype and say, what does this person appear as and what syndrome do they have? And you'd say, well, look, even if I didn't number them, you guys, the fact that these 3, as long as I told you they were paired up, you said there's 2 big ones and 1 little tiny one. That's XXY, right? That person's going to appear male because he's got a Y chromosome, but this is Klinefelter syndrome, 47XXY. All right? Now, an interesting question that may have crossed your mind or may not have is, well, I've mentioned certain ones, right? Certain examples of trisomies, like with the autosomes, Down syndrome, Edward. With the monosomies, there really aren't any autosomal ones around. With the sex chromosomes, we now understand, but I want to know, why don't we see any other trisomies or monosomies? Specifically, autosomal ones, like trisomy 3 or monosomy 22. The thing is, it's so bad, they cause such severe problems during development that they always lead to spontaneous abortion or miscarriage. Guys, whatever you think about abortion, this has nothing to do with the ethical debate. The point is, when someone has a miscarriage, many times, if permission is given, they'll do a genetic analysis on that embryo to find out what happened. It really helps us to learn. And more often than not, they do find a chromosomal abnormality. And they say, you know what, this is the way things work, and it's biology. We can't do anything right now about it. This zygote was created. It was able to grow to a certain point, but the chromosomal problems at a certain point were so severe it could not continue. So sometimes a miscarriage, I'm not saying it's good, it's traumatic, but it's nature's way, if you will, of saying this is not going to happen. I only mention this, a lot of women feel guilt when they have a miscarriage. A large percentage, not all of miscarriages, are inevitable. They will happen because this embryo or fetus is chromosomally defective, for lack of a better word. Okay? So we say they're embryonic lethals. The embryo dies and it gets miscarried. No one is ever born with these syndromes, all right? But this is what I need you guys to realize. It's definitely important to understand that in general, zygotes for all the potential trisomies and monosomies are created with equal frequencies, right? So, because, there's no reason during non-disjunction why chromosome 21 preferentially this happens to. It happens to all of them. So there are zygotes that are trisomic for number 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, etc. in the same frequency and monosomic. It's just that they fail to result in any viable human being, so we don't ever see them. The only ones we see are the ones I mentioned because they're either not bad at all because there's sex chromosome problems, which we know your body can, quote, fix, or they're things like Down syndrome, which is not that bad, meaning it doesn't affect genes that don't allow you to exist, to live, right? We see with Turner and Edward syndrome, Patau and Edward syndrome, these are some of the most traumatic ones because they're really bad, but not so bad that they, lead to spontaneous abortion all the time and get it over with. Individuals can be born with these very severe conditions. All right? So I found a karyotype of, this was a miscarried embryo, and they wanted to see what happened with it, and they did a karyotype. This would have been a girl. And the problem was that there were 3 number 16s here. So this is trisomy 16. I think you're clear about this, but realize if I showed you this karyotype and it's sad, and I don't mean to be morose, but this could never be a viable human being. This has to be miscarried. It's an embryonic lethal. That's probably the best way to say it without getting too upset. So it's trisomy 16, 47 plus 16, embryonic lethal. But it's interesting to see, right? But no human being walking around will ever have a karyotype that looks like that. All right. Now, I could have left it at that, but I have to stir up trouble. There is one other question that a student asked me once, and I said, that's a good question. And we don't fully know the answer. But here's the deal. With the sex chromosome trisomies and monosomies, I say if you think about it, it's not even clear why an individual, for example, with Turner syndrome should have any phenotypic abnormalities at all, right? Because think of the logic. I'm telling you the proper dosage of the X is 1. I have 1. I'm a male. If you're a male, you have 1, right? Nothing happens. We don't make any bar bodies, and everything's fine. Well, a Turner syndrome individual has 1 also. Remember, they're XO. They have the 1. They don't look male because they don't have the Y chromosome, but they have the 1. It doesn't get turned off. Why should there be a problem at all? It's a very good question. Well, it's not fully understood as I said, but what it seems is this, that there are a small subset of genes necessary for proper development located on the X chromosome that are not inactivated when a bar body forms. And additionally, there's evidence that some genes on the X chromosome are also reactivated at puberty in women during gametogenesis, which means the making of eggs, which would account for the sterility problems. But interestingly, guys, in mice, the equivalent of Turner syndrome, we don't say a mouse has Turner syndrome, but XO individuals have no phenotypic problems whatsoever, implying that we don't understand. Maybe these syndromes are peculiar to humans. And we don't have to go through it all, but the same logic could be applied to, say, a Klinefelter guy. XXY. Well, one of them gets turned off, just like in a normal female. Why should there be problems? So something has to be up with the fact that some of those genes on the X chromosome, even when they're turned off, right, even when the X chromosome is inactivated, there must be some gene activity at some time, and the lack of that leads to the problem. Because remember, even if you're got 10 X chromosomes, which would never happen, but if 9 of them got shut off, here we have a dosage compensation mechanism. So one of them is always functioning. So it's the lack of the additional functioning that causes the problems, it seems. It's still a little confusing. We don't completely understand it. All right, you guys? All right. Well, that's what I have to say about aneuploidy. It's a very interesting topic, and we're going to see, if you go in order at the next session, what happens when we change complete sets. We call that polyploidy. I'll see you then.